Welcome back to the deep dive. Today we're shifting our focus. We're looking inward, way, way past the scale we normally think about, into a world that, well, it fundamentally controls things like our air, our food. We are diving into the foundational science of microbiology. Our mission here is to get a quick handle on the incredible scale, the history, the importance, and, the basic classification system that really defines life on Earth, especially how structure relates to function for these tiny organisms. Okay, let's unpack this. Maybe starting with a bit of cosmic perspective.
Yeah, that's a great place to start. Because when people think vast scale, they look up, right, to the stars. Astronomers reckon there are, about 1 times 10 to the 24 stars out there? It's sort of the definition of huge.
Astronomical.
Exactly.
Yeah.
But here's the thing. If you look down right here on our own planet, the numbers, well, they make the cosmos look almost manageable. Take viruses, for instance.
Yeah.
The estimate is around 1 times 10 to the 31 viruses, just around.
Whoa, hang on. 10 to the 31.
That's 10 million times more viruses than stars. Staggering.
Okay, just trying to visualize that. The source material mentioned, like if you could stack them end to end.
Yeah, get this. They'd stretch about 100 million light years.
100 million light years. Isn't Andromeda only like 2 point something million light years away?
It is. So this chain of viruses would be about 43 times further than the distance to Andromeda. It's just...
Yeah. That's not a population. That's, like you said, a microscopic universe in itself.
It really is.
Yeah.
And bacteria aren't far behind. In the oceans alone, we're looking at maybe 1.3 times 10 to the 29 bacteria. Again, way, way more than the stars. 100,000 times more, roughly.
Just in the oceans.
Just in the oceans. And think about this. You scoop up a teaspoon of regular garden soil. You're holding about 1 billion microorganisms. 1 billion.
Wow. Okay, the scale is clearly almost incomprehensible. So if we're dealing with numbers like that, let's define our terms. What exactly counts as a microorganism? Small feels a bit vague now.
It does, doesn't it? Formally, a microorganism is an organism that's too small to be seen clearly with the naked eye. Generally, the cutoff is around 1 millimeter or less in diameter. And that size limit is important because, it means we need tools, microscopes, specialized techniques to even see them, let alone study them.
But size isn't the whole story, right? The source material emphasizes that.
Exactly. It's necessary, but it's not sufficient. What really defines them is, like you said earlier, structure and function. How they're built, what they do.
Because doing something as huge as, say, creating our atmosphere requires some seriously specialized machinery inside those tiny cells.
Absolutely. Microbiology is biology, yes, but the unique properties of microbes, their sort of alien lifestyles in some cases, mean we need unique ways of looking inside.
Which brings us to the basics we need to know, like macromolecules, nucleic acids, proteins, carbs, lipids.
Right. And it's not just about memorizing a list. It's about understanding that in the microbial world, how these molecules are put together dictates everything they can do. Structure determines function profoundly.
And where they can live, too, presumably. We talk about prokaryotic versus eukaryotic cells, but why should, say, a specific lipid structure matter?
Oh, it matters immensely. Lipids make up the cell membrane, the boundary. And some microbes, especially in the domain archaea, have really unique lipid structures. These structures allow them to survive in places like boiling hot springs or incredibly salty water, places where our kind of cell membranes would just disintegrate.
So their membranes wouldn't, melt or fall apart?
Pretty much. Their structure is adapted for survival in those extreme conditions. Understanding their cell walls, their membranes, it's fundamental to understanding their success in these, frankly, hostile environments.
Got it. So those chemical differences are literally life or death at that level. Okay, that connects nicely to function. If we connect this to the bigger picture, what are these trillions upon trillions of microbes actually doing out there?
Well, they're essentially the planet's engineers, the unseen workforce. You can trace modern life back to a pivotal moment about 2.4 billion years ago, the Great Oxidation event.
Ah, the cyanobacteria.
Exactly. Cyanobacteria figured out a type of photosynthesis that releases oxygen as waste. They were the first to do it on a massive scale.
And they basically terraformed the planet, releasing all this oxygen, which was toxic to much of the existing life, but paved the way
For oxygen-breathing organisms. Like us, it's arguably one of the most significant events in Earth's history, driven entirely by microbes.
Incredible. What else? Nitrogen fixation comes up a lot.
Yes, absolutely critical. Nitrogen gas makes up most of our atmosphere, but it's unusable for most life in that form. Only certain bacteria can perform nitrogen fixation. They convert that atmospheric nitrogen gas into organic forms, like ammonia, that plants and other organisms can use to build proteins and DNA.
So without these specific bacteria, the whole nitrogen cycle stops. The food web collapses.
Essentially, yes. They are indispensable. It's not an exaggeration. They're also the planet's recyclers, constantly breaking down dead organic matter. Imagine if they didn't.
We'd be buried in dead stuff.
Pretty much, they keep nutrient cycling.
And it's funny, alongside these massive planet-shaping roles, they also give us beer and cheese.
Huh. Yes. It's quite the contrast, isn't it? Beer, wine, chocolate, cheese, yogurt, all rely on microbial fermentation. They sustain global ecosystems, and they hang out in the dairy aisle.
It really highlights this duality. Life-giving on one hand.
But also sometimes life-taking. Which brings us to the less pleasant side.
Right. Infectious disease. The numbers are stark. Something like 16 million people die every year from infectious diseases globally. That's 1.6 times 10 to the 7 deaths.
It's a huge global health burden. And here's the deep irony. Many of the tools we use to fight these diseases, vaccines, antibiotics, are themselves derived from microbes.
Fighting fire with fire, then.
Precisely. We're using microbial products or processes to combat harmful microbes. Penicillin, famously, comes from a mold. Many other antibiotics are bacterial products. But what's really mind-boggling is how little we actually know about the microbial world as a whole. Those disease-causing microbes, the pathogens we study so intensely, they represent less than 1% of all microorganisms on Earth. Less than 1%.
So we focus almost all our attention on this tiny fraction that harms us.
Well, the other 99% plus, at the vast majority, remains largely unexplored. We know they're out there doing things, but we haven't even identified most of them.
And just like trying to count stars, figuring out the total number of microbial species, bacteria, archaea, viruses, fungi, protists, it's still very much an active area of research and debate. We don't have a definitive catalog.
Not even close. And that uncertainty is why classification became so crucial. Early on, scientists tried to fit everything into plant or animal, which obviously didn't work for microbes. They relied heavily on what things looked like. Shape, size, staining properties.
But looks can be deceiving, especially with microbes.
Very deceiving. The real game changer came with molecular techniques, especially looking at genetic material. Carl Woese's work in the 1970s was revolutionary. He focused on sequencing ribosomal RNA, rRNA. It's a molecule essential for protein synthesis, so all cellular life has it, and it changes very slowly over evolutionary time.
So by comparing the rRNA sequences,
he could measure the evolutionary distance between organisms much more accurately than just looking at them. And this led him to a radical conclusion. He proposed the three-domain system: bacteria, archaea, and eukarya.
Right, this overturned the older five-kingdom model because Woese showed that the prokaryotes, the simple cells without a nucleus, weren't one group. They were two fundamentally different domains.
Exactly. Bacteria and archaea might look similar under a microscope. Both are prokaryotic, no nucleus. But their rRNA sequences showed they're as different from each other as they are from us eukaryotes.
And this explained those extremophiles, right? The ones living in boiling acid or deep-sea vents. Many of them turned out to be archaea.
Precisely. Their unique biochemistry, those lipids we talked about, reflect their ancient, separate evolutionary path. They aren't just weird bacteria. They're a whole different domain of life.
That shift in thinking is huge.
Yeah.
So for someone learning this, the practical skill isn't just memorizing bacteria, archaea, eukarya. It's about being able to look at the characteristics of a newly found microbe and figure out where it fits.
Yes. Can you determine if it's a bacterium, an archaeon, a fungus, a protist, maybe even a virus which isn't cellular? What are the key structural and metabolic clues?
Because knowing its classification tells you a lot about its potential role, its importance to us, right?
Absolutely. Is it cellular? Does it have a nucleus? Eukarya, like fungi, protists? Or is it prokaryotic, bacteria, archaea? This tells you about its basic biology, how it might reproduce, what environments it could tolerate, maybe even how it might respond to antibiotics. Structure, classification, function, they're all linked.
That connection to health, the environment, biotechnology, it really runs through everything in microbiology. It's not just abstract knowledge.
Not at all. It's incredibly relevant.
Okay, so let's quickly recap the journey we took. We started with the almost unbelievable scale of microbes literally outnumbering the stars.
Mind-boggling numbers.
Then we saw their absolutely essential role in shaping our planet, creating oxygen, cycling nitrogen, driving ecosystems.
Planetary engineers.
We touched on the duality. They cause devastating diseases, yet they also provide the cures and many foods and products we rely on.
The good and the bad, often from the same source.
And we landed on how we try to make sense of it all through classification, thanks to breakthroughs like Woese's three-domain system based on molecular evidence.
Which brings us back to that vast unknown. And this raises an important question, something for you to really think about as you study this field further. If less than 1% of all microbes cause disease, and we're still just mapping the known domains, what about the other 99%? This massive, largely uncharacterized majority living in soil, oceans, even inside us. What potential does it hold? What new biochemical pathways? What undiscovered enzymes? What solutions to problems in medicine, environment, or technology are hidden within that microbial dark matter? What are we missing?
That really is the frontier, isn't it? What else is out there waiting to be discovered?
It's potentially transformative.
A truly massive world hidden in the microscopic. Well, thank you for joining us on this deep dive into the foundations of microbiology. Keep asking questions, keep exploring,